Good afternoon, everyone. I think we should get started. Thank you all for showing up here in person. Deeply appreciated. So welcome to today's, this is, I think, the concluding episode of the NIH Research Festival and also the third Wednesday afternoon lecture series in the 2024-25 season. For those of you who don't know me, my name is Anirban Banerjee. I am a PI in the intramural program of NICHD. I'm also a member of the ANHPI Federated Asian Network Group. Today's nomination was supported by the NHPI community, and we deeply are grateful to the NIH leadership for hosting Manu Prakash. And it's a distinct pleasure and honor to introduce Manu, who comes to us from Stanford University, where he is in the Department of Bioengineering, but also a senior fellow in the Woods Institute of Environment. And he's also a senior fellow in the Center for Innovation in Global Health in Stanford School of Medicine. So I think I perhaps could just say that Manu is a rock star scientist and be done with my introduction. But the problem is that some of these words and phrases have really lost their meaning because of overusage. So I have to elaborate a little bit. Manu got his undergraduate degree in the Department of Computer Sciences at the Indian Institute of Technology at Kanpur. This is the department that trained the trainers of people who brought us Google. So getting into the department, I think, is in itself a qualification. And then on, Manu went on to the very famous Media Labs at MIT to do his PhD. He was a junior fellow at Harvard and then started his career at Stanford and has risen through the ranks. Manu's scientific interest is in understanding computation in biological matter. He's also deeply interested in innovating new tools for studying non-model organisms with a focus on life in the ocean. And I think he's most well known for distributing frugal science tools for really sort of democratize access to science and diagnostics of deadly diseases. He's best known for his invention Foldscope. But what is very evident when you look at Manu's choice of science problem is he really exemplifies the free scientific mind who looks at the world as a treasure trove of problems and is then able to go and dissect them and really analyze problems and solve problems that make a huge difference to human society. For those of you who can see, on my T-shirt is an image of Tagore, who's one of the greatest poets of the Asian continent. And the introductory line of one of his most celebrated poems that says, "Where the mind is without fear and the head is held high, where the knowledge is free," and it goes on to say, "Into that heaven of freedom, my father led my country away." I think nowhere is the path between science and a stronger society more evident than the likes of Manu Prakash and his research. Thank you for coming to the NIH, Manu.
Thank you. I don't know where to begin. I hope I don't disappoint you. I am absolutely delighted to be here. It's an absolute honor. This will be far more informal, so please feel to interrupt me anytime. We have some amount of time with us. I have 200 slides. I don't have to go through any of them. We can have a conversation. I brought some toys with me. We can spend some time, if you're really interested, to actually use them and play with them. And we'll kind of do this in a manner of hopefully as a conversation. So I think before I begin, one of the important things I want to get out of the way is I'm a scatterbrain. That's depicted in a breadth of work that we do, and it's very important to actually first acknowledge, because I'm known to not reach the end of my talks, is I don't work alone. I work with a remarkable group of people, and only a few of them are here in these photographs and many lab alumni that now take the culture and philosophy of the science that we do around the world, but also an infinite number of people out in the field that have inspired the problems that we have picked on, many community health workers that held our hand, truly telling us the directions that we should go. And many of the patients that have actually changed and asked us really good questions that have enabled us to go in these paths. So without many of these individuals, I don't think we'll be able to even frame the right sets of questions to begin with. There is one message that I want to communicate across in all of this work, which I think connects a lot of things that I think about, which is the idea of magnifying curiosity. You know, it's the seed of all of science. And I think it's a false debate when we start talking about applied work and basic work. Often enough, we all in our lives and scientific lives juggle through it. So in some sense, this has been one of the goals that we've taken on, and not just thinking about in the scientific endeavor itself. Curiosity has a role to play much broader. And I think it's important for us to be thinking about sharing that sense of curiosity that we all get to both experience and practice, but to actually share that. So although there are incredible number of my own scientific curiosities that I've spent infinite amount of time working on, I'll give you a tiny glimpse of it. The talk really will be about how do we move forward and share that sense of curiosity with the broadest sets of communities. So, you know, much of what we write is published and written. Many of you can take a look, but I'll give you a glimpse of some recent preprints that we have posted that I'm hoping will evoke your curiosity. We're also known to have a zoo at our lab, so we have around 25 different organisms that very few people even try to grow. But I'll show you they're collected from places around the world. This is just the last 5 years of expeditions that have allowed us to collect and actually observe, for some of them, for the very first time, these sets of organisms. And, you know, while I have this slide, I spent a lot of time at sea, more recently in the last 5 years because of the context of climate and what's happening to our planet. And in fact, much of the cell biology that we know nothing about is what's driving the adaptation to the planet. You know, some of these sets of environments and ecologies both make me wonder how little we actually know. And I'll give you an anecdote of that just on the side. I very recently came back after 35 days at sea, sampled roughly around 150 million liters of water. It's a lot of water. So, you know, kind of going from Eppendorf's to 150 million, but it's still a drop in the ocean. And in our hands, I ended up finding a cell, one single cell in 150 million liters of water that has actually only been seen once by humanity in 1898. There is only one drawing of it. It's never been photographed. And you drop your net, and if you're lucky enough, outfind you. It represents a tree of life that all we know is this one painting, and that's about it. So I think, I mean, you know, in that context of, come talk to me, come for coffee if you actually want to see that first photograph. I don't have it in the talk here. But I think I'm just sharing the sense of wonder that we all live with, and just the moment we step out of the lab, how much we actually don't know is just astounding. And so I try to spend as much time as I can. I mean, and again, much of that can be also said for pathogens and parasites, as well as much of the biology in our ecosystem. So let me give you a flavor of some puzzles recently that we've been thinking about. The object that you see there is, I think it's the cutest animal on the planet. It's not a tardigrade. These are sea cucumber juveniles. So you've all, how many of you have touched a sea cucumber or eaten a sea cucumber? Wow, so many people have eaten a sea cucumber. Wow, that's fantastic. They don't taste that good. What's remarkable about echinoderms is that they're called echinoderms because they build their body plan with rocks. And what you're looking in polarization is essentially single crystal of calcite, that a type of cell that we discovered, a sclerocyte, builds a single crystalline structure. And now I've taken these sets of things, these poor juveniles, then went through an X-ray And we've been able to map the entire topology and the algorithm for how does a cell, as a sculptor, build a single crystalline architecture of all these sets of shapes and forms. And you might ask, where is this shape encoded? You should read the paper. It's for the first time allows us to bring cell biology to the problem of biomineralization. There are microtubules, there are motors involved, there are vesicle pathways, and really interesting parallels between how neurons and much of our vasculature shapes itself, but connected to the problem of biomineralization. So anyway, this is on arXiv. I'll give you another example of a puzzle. This I discovered with a foldscope, which I'll talk quite a lot about. In a swamp, in a pond, I have a tradition of taking all the new students, including postdocs, to this swamp. I have not lost a student as yet, but often enough we find rarities that are just sitting right in front of you. So I'm just going to play this video. This is real time. The length scale in that is that the little blob that you're seeing on the top is only 40 microns. But I forgot to mention this is a single cell that's transforming right in front of your eyes. So any of you think about, membrane mechanics, here is a cell that's generating a neck that's as much as 60 to 70 times its size within a second. And there is a cytoskeleton associated with this structure that you can see on the right. It's one of the most beautiful cytoskeleton structures. It's a ciliate. And what we were able to discover in this system is the first example of origami, the art of folding paper. And the microtubules are patterning the membranes in a manner to fold it in this tiny little structure. So that's the folded state, that's the unfolded state. And there's a really beautiful mathematical analogy of there happens to be these topological singularities that control the deployment of this cytoskeleton in and out. And this organism, if you were the size of this organism, you should be quite scared because it's going to snatch you like a fishing line, and it uses it for feeding. And again, unless we look, I would have never just dreamed. I think human imagination is actually quite poor, and often enough it's just being able to visualize these sets of systems gives us these sets of insights. Another system which is more close to pathogens. If some of you think about microsporidia, we recently cracked the puzzle for how a tiny little spore, without any mitochondria or any other known sources of energy, shoots out these massive polar tubes. So I'm just going to slow that down a little bit, and you'll see these remarkable architectures that build and arise. And one of the threads around this is being able to observe these processes is the time when you get to ask the right question. So I think I've been obsessed with this notion of to truly ask the right sets of questions, we should spend time observing life. And you're going to see this pattern over and over again, both in the context of diseases and in the context of basic science questions. I'm going to close this with just one more example from Flatland. This is a system that we've been studying for a very long while in thinking about the origin of behavior, especially in the context of animals where neurons had not actually evolved. Because in the metazoan tree of life, what happened in the Cambrian time is all of these sets of systems have neurons to begin with, have muscles to begin with. But at the base of the tree of life and animals are a few, only 2 known phyla that have no neurons and no muscles. So what you're looking at is this bizarre animal that I think while I wasn't paying attention, half of it decided to walk away from its other half. I'm hoping I'm not as incoherent that one of my half decided this is asexual division. All happening with no neuronal kind of a context, all driven by cilia. And it's led us to think very deeply about the idea of flocking dynamics that occurs in birds to actually be applied to ciliary and cilia mechanics and cilia control. I'll give you one last example of the strangeness of our world. I know many of you think about single cells, so you might wonder how cells regulate their volume. And one of the puzzles that's been is the fact that what is the capacity? What if I told you that a cell could dilute itself 6-fold in its volume within 10 minutes? So many of the biochemists in the room should worry, because that's going to dilute every enzyme, every possible thing that you think about. And right here is a cell. We found this off the coast of Hawaii, around 500 meters deep in the water, that actually uses inflation as a mechanism to migrate 500 meters every 7 days. So it's a single cell, and we have an instrument that we've built that allows us to travel these journeys, and you can see these massive inflation events. And it's technically non-motile, but it's actually uses aquaporins and being able to flood water into the cell to change its buoyancy to go up and down. And it turns out in this context, the cell cycle is actually deeply connected to this set of a behavior. So, lots of fun papers that you can read, but I decided not to talk about any of this because many of these things you could read. I want to spend a lot more time on this philosophical framework for how do we go about bringing observation at the heart of science and bring that to as many number of people, because primarily, unless we do something like that, we might be in a world where we are going to be losing both scientific knowledge, sets of species, and in the context of health of the planet, things faster than we can even discover them. So I'm going to transition and tell you a lot more about this philosophical framework that we've been calling frugal science, and how do we, beyond the science that we do in our labs, how do we go thinking about taking the kinds of tools that are now possible to develop in hands of almost, say, every kid in the world? And this is an endeavor that happens with communities at the center. And before I begin, I think I want to share the hard truth with you so that there are reasons I lose sleep at night, and I want you all to lose sleep at night. I teach this class called Frugal Science. I have students that come from around the world that take the class, and this is Marwan, who's a student. He lives in Sudan. He's been in my class 2 years now in a row, and we've been working on many things. And this was just before the war broke out in Sudan. And we were having this set of a conversation, and he said to me, Manu, what you're trying to say is this idea, we have this very old Sudanese saying, that don't make a pen that doesn't eradicate ignorance and shame. Pen was a symbol of technology for him, and he was really talking about access. Is this idea that what is the point of this wonderful world of technology that we build around unless it reaches people? And when we say people, that implies people around the world and the haves and the have-nots simultaneously. And I think that stuck with me for a long time. And it's framed in this context. And I'm going to tell you sets of, first, the depressing part of why we all need to care about that. But then secondly, what each one of us as an individual can actually do about that problem. So first of all, let's start with education. This is a photograph that I took. I actually went to a school that looks something like this. And often enough, if you think about around 2 billion kids on this planet, roughly 1 billion of them live in a family that would be qualified in many sets of things around under the poverty line. That's literally half of the entire future workforce in some sense. COVID hit. Many of us saw what happened to education in that context. This was the virtual classrooms of the world, and UNICEF has kind of estimated this roughly around 500 million kids were completely cut off from a context of education. And again, we, even in this country right here with all the sets of resources, we have dramatic challenges in thinking about engaging in a curiosity-driven approach to science. But we have a much larger challenge when you start amplifying and asking this set of question from a much larger perspective. In parallel, while we face these sets of challenges, we are facing a remarkable time in our lives, which hopefully we will get to tell the next sets of generations about some of the largest biodiversity loss that we're seeing. This is a paper from Germany documenting many of the insect losses that they've actually seen. You can go back to health. There's a lot of work that we do in the context of health, and you'll see that happens to be a clinic. I know many of you work on malaria. This is the harsh reality of malaria. And one of the things that starts to happen is that we are not able to deliver the kind of care and diagnostics in most settings around the world. And, you know, often enough this is driven by the sets of distances, the sets of challenges. There are certain sets of criteria that you have in control. This is one of our field sites in Madagascar. 12 hours is an average road walk to get to a clinic to begin with. And that kind of has been bothering us for a very long time, that unless technical tools and technologies reach the sets of communities, we might never be able to tackle some of these sets of problems. And, you know, I think malaria I'll talk quite a lot about. There is a lot of concern in thinking about what's happening and the evolution of malaria out, and just its spread per se, but also new sets of vectors that many of you probably heard about. There are some urban mosquitoes at this point. They're all deeply connected to the ecological and environmental sets of changes. And then finally, I think another important perspective, if not all of this was already worrisome enough, one of the largest ecosystems in the biosphere on our planet is dramatically changing. So if you just do the calculation, 70% of the surface of the planet is water and ocean, but actually more than 90% of the living biosphere is also the ocean. And an important aspect of this is, because of the cellular life and very fundamental mechanisms that we don't understand are operational. So if you look at this curve, you can see the CO2 that we've been pumping on the top side, and in the carbon budget there is actually a curve down below that's called the ocean sink. The ocean has been absorbing proportional amount of CO2 from our atmosphere, between 30 to 40%. As we keep increasing, the ocean keeps doing more, but we have no idea in the context of the tipping points associated. Often enough, we don't even know who lives there to begin with. Forget about how this largest carbon sequestration technology on the planet actually works. And again, goes back to very fundamental basic science and our capacity to even observe our planet and ecologically monitor our planet. So I hope you find this concerning. I lose a lot of sleep, and I think one of the things that I often worry about is this narrative sometimes that is technology the answer to all of this. We should not worry about it. There's lots of people, and eventually we'll end up technical sets of solutions. And, you know, I would say, arguing COVID, a lot of those sets of things did turn out to be true, but we also know the things that did not work so well. And often enough, I am drawn back to thinking a lot about access and inequality, and primarily using this framework from the very beginning of development of technologies. So rather than thinking much later about how am I going to scale the sets of tools that you might care about, how do we do that from day one? How do we really think about, even before starting a project, can you really build access into the design of the sets of projects? So, you know, I didn't come here just to tell a depressing story. I do believe that we can, and we have as humanity, a capacity to change and dramatically change the course of our trajectories, both in health and in environment, and I think they're deeply connected. It requires two key principles. One is truly building planetary scale measurement tools. You know, without that, we have very little feedback on the sets of drugs that we put out there, the sets of perturbations that we do, whether they work or not. And secondly, we don't have enough scientists around the world to even be able to tackle. We don't have enough doctors. And we've relied on community health workers and many broader sets of community sets of programs. And at some point of time, we have to come to the table and agree that there has to be an inclusive approach of amateur scientists and individuals that have a capacity to engage in these sets of problems to play a bigger role. And that's planned out in community health, but actually has not been taken on in many other sets of places. So, you know, I think the principle that we'll talk about is frugal science. How do you engineer for cost and performance in a careful manner that you can really deliver sets of solutions at cost. I'm going to give you a few sets of examples. I'm also looking at the clock and also realizing my talks usually go exponentially faster as clock goes by because I have so many things I want to tell you, but the most important thing is the philosophical framework for why we do this. So I apologize in advance if I'm going to skip through and rush through many sets of things. So let's start with something from the past. How many of you recognize this object? That tells your age, which is good. That's Sputnik. When Sputnik was launched, we had no idea how to track satellites. There was a brilliant man, Fred Whipple, from the Smithsonian, and he thought, what if we allowed citizens around the world with tiny little telescopes, cost $10, $20, tens of thousands of people to watch the skies, and for the next 60 years, while DARPA and many of the other defense agencies of that time were building these automated sets of telescopes to do satellite tracking, for 60 years, it was this network of a community. They were called moon watchers. And all our astronomy data, the first supernova, majority of the Sputniks that were launched were actually seen by these community members. They were doing quantitative data collection. And I think this has been an inspiring story for us when we started thinking about full scope per se. You can buy these telescopes still on eBay if you're an aficionado. Most people don't know what they are, and so they go for a pretty cheap price. So is there a parallel? If you really start thinking about, I think the citizens, the community health worker are the largest citizen science organization in the world today. They don't get paid. Many of them are in volunteer sets of scenarios, but they also put themselves on the front lines of diseases that we all deeply care about. And I often think about, this is a picture from Last Mile Health, of an individual that's about to head out into places where they are detecting and looking for Ebola, and she's carrying a backpack. And often enough, the sets of tools and technologies that we build, some of that is here. I'm often thinking about what goes in that backpack. How do I amplify and think about the kinds of things and the science you can do out in these sets of field conditions? We spend a lot of time in the field, and I think it's really remarkable that many of these individuals teach us along the way. And I remember this very specific story of meeting a doctor in Uganda. I was a young engineer, scientist, excited, and I'd taken some of the tools. I'm not very shy. I really always want to share sets of things. And he told me, you know, Manu, you should come back when you can truly do diagnostics under a tree. And what he was trying to tell me is that, you know, it's very important to deeply understand the context and the sets of challenges and problems. And I think I've taken that to heart. And a lot of work that we do has been inspired by the feedback that's given by many of these community members that actually engage. There's a whole list of tools that I could talk about that we've been building. I'm just going to touch upon a few of them. They range from diagnostics to science education to environmental surveillance, tracking mosquitoes. I don't think I'll have time to touch on each one of them, but we'll spend a little bit of time on a few of them. So let's think a little bit about, I think, you know, COVID probably, the only thing I'm going to mention here is manufacturing has been a key principle for us when we're thinking about medical device design. There's a whole bunch of innovations that many of you worked on in this set of a context. And one thing we focused on is building a manufacturing framework. So for one thread that was important in medical sets of devices is, although we could invent many of them, there is very little framework to be able to do high-quality device manufacturing. We partnered with actually an airplane engine manufacturer in India. They had never touched a medical device before, but I knew they had the capacity. I mean, if they're making a sophisticated device like that, we took a team from them, trained them on biomedical engineering from scratch, built a regulatory program with the same sets of tools that are used here, and actually that led to a product being built by that division from scratch. And I think it was a really enriching experience in thinking about what does it mean to build manufacturing pipelines from scratch, because of course all of these sets of devices and designs have existed for a long while, but we haven't taken the step and the leap to be building a much more robust manufacturing pipeline. And this manufacturing will show up a couple of times in my talk. Okay, so let's start with a few sets of tools. How many of you have used a foldscope? Okay, so that's not all the room. I have enough foldscopes for all of you, so if you stay out for coffee, feel free to take them away. This is what that tool actually looks like. I think one of the scenarios is thinking about, we were thinking about microscopy, and we came upon this notion that could we manufacture microscopes like printing books or newspapers. That led to this tool, and I think maybe one of the threads here is when you think about microscopy and manufacturing, paper doesn't come to mind, and that was exactly what we wanted to do. So this was the very first blueprint of Foldscope version 0.0. When we shared this on an archive paper, it was kind of interesting. I mean, our goal was, can you do 700 nanometer resolution imaging at a price point of $1? And we spent 6 months solving a fat lens equation. We figured out high curvature lenses can actually be used for very high resolution imaging if you follow a certain sets of rules and principles. That led to the microscope. We published it. I was very proud of that paper. This was the first paper I wrote as my new lab at Stanford. But nothing changed in the world the next day that I woke up. And, the world was still exactly the same. And that really hit hard. And ironically, something else happened, which is I didn't realize that Bill Gates reads archive papers. And he posted this tweet, and there is something very particular I want you to pay attention. It says, $1 microscope, ? Interesting. And I was stuck with that question mark. Like, what does that mean, ? Does he not believe that we have done this? And, he's a businessman. He understands what he does. But it spawned an idea. It made me realize that no inventions that we do, unless we take the ownership of bringing them to communities that we care about, none of that matters. It matters to some people. It might take my lifetime. And at that moment, we decided we are going to ship 100,000 Foldscopes to people around the world. We got messages. I just posted that on the website. And we literally built a manufacturing pipeline in the lab. We realized we were just 3 people in the lab at that time, built a robotic factory in the lab, completely autonomous manufacturing. And I spent $250,000 of my startup money just shipping microscopes. No question asked. I know some of you are in the room that got that first batch. I made deep friendships. And what we were doing accidentally is we were building a community. It was not about the microscope after all. It was about passionate people engaging in the process of science with no transaction. There is nothing we wanted from them other than explore their own curiosity. That created a network around 150 countries. And today, at this point, there is 2.5 million users of Foldscopes around the world. There are 60 distributors. We've scaled this set of a program, and we're now ramping up for the next round of scale-up. And it's been remarkable to see, if you just really feed curiosity, what people are capable of doing. I love this photograph because these are the parents that are not sharing their microscope with their own kid. You have to be that parent. If you care about bringing science to people, you really have to embody it, that you live it and breathe it. And we all do. But often enough, when we are in a public domain, we're much more shy about how we would approach. At this point, Foldscopes are used. I mean, this is just the kind of data that comes out of Foldscope. One of the things that we've been thinking about is a software platform that supports it. Now there are AI layers on top of this. And it's become one of the largest microscopy community that's distributed. We have users in Iraq, Afghanistan, Congo, all parts of India, Alaska, Nebraska, any place that you can throw a dot in the world, there are users around. And I think one of the passionate things that's been is that our goal has been empowering many of these individuals that then drive the programs. So we don't control. It's not a hub and spoke model, completely distributed. People build the sets of programs that they care about. And you are included in that age group. So, and I think one of the threads to think a little bit about would be is who is a scientist? Who do you think actually can make a discovery? That's a question that we should ask ourselves every time, because it's been remarkable to see. I've only written one paper using a foldscope. There is roughly around 700 publications at this point. Many of them come from communities and groups around the world studying organisms that I would never have access to. So I think, it's kind of changed my perception of how should we do science and who do we engage in that process. There are thousands of examples, and since I'm running out of time, I'm not going to spend any time on this, but kind of one of the frameworks is they're used in an agricultural context, animal health, human health. There are chickens that use foldscopes. This is actually a serious picture of a sailor. He happens to be the youngest sailor to cross the Canadian Arctic around a frozen time while imaging and spending time at sea. There's biodiversity maps, and maybe I'll just highlight one person in here. That's Mo Pandirajan. He runs a rural school. When I first met him 7, 8 years ago, now this one single person has actually trained 95,000 students all across India, 12,000 teachers, just one person. And kind of one of the things that happens is that many of the Pandirajans are in this room. And often enough, you ask yourself a question that, oh, what can one person do? This kind of an amplification has been very powerful for us to think about. That's Iraq, Syria, Lebanon, Puerto Rico. Lots of animals are diagnosed using a foldscope. And I think one of the things that I've been thinking a lot about is how do we now translate many of the principles that we learned with this kind of an approach to other products. So I think I told you a little bit about microscopes. I'll just briefly say some things about diagnostics. I don't have to tell you how big a problem diagnostics is. We started first thinking about molecular diagnostics, and then I'll talk a little bit about imaging-based diagnostics. I think our manufacturing capacity for molecular diagnostics is very limited. We asked ourselves this question: could we truly build a factory in a box for distributed manufacturing of diagnostics? Every enzyme, every chemical, everything built in a small-scale facility, all the way from both the molecules and the gadgetry, all put together in a manner to build a distributed molecular diagnostics factory. This is work in progress, but I think we've made all the pieces in place, and kind of one of the powerful things has been, this is called SnapDX. It's essentially a way to do a lamp assay with no electricity, just hot water. So we kind of joke about, if you can make a cup of tea, you can do diagnostics. There is a lot to talk about in terms of the physics of how this works. It's inspired from the manufacturing of a ballpoint pen. And I think the only thing that I'll just mention is, during COVID, we were able to go from all the way from an idea to the first clinical trial and usability trials in a short period of time. And soon we started realizing that, yes, we can actually do this. But the framework then shifted from thinking a lot about not the diagnostic test as the product, but the actual manufacturing as a product. And kind of just for aficionados in the room, this is us testing SnapDX out in Liberia. And you can see that as a diagnostic tool. I'm actually curious if any of you can spot another very important health technology that is hidden in this photograph. Not the diagnostic test. You can just shout it out. Yes? Huh? Phone. That's interesting. Another one. Actually, that's a good one. Any other one that you see? Motorcycle. Exactly. Motorcycle is a very empowering health technology. Of course, we didn't build it for that purpose, but much of the ambulance system in countries around the world would shut down. And again, in the framework, if you can share the sets of technologies, remarkable people engage and build services and programs on those same sets of tools. So that's the philosophy that we have taken, is that build the engine and allow other people to build on top of it. And I'm going to give you 2 examples for that. So first of all, just going back on manufacturing for a second, we've been able to just build the entire FPLC pipeline in a frugal manner. We can synthesize most of these things. I don't own the truck, but I have a truck. And we're literally building this entire pipeline that we have in the lab in a truck. And then we have a site in Senegal that we're putting this with a partnership with Institut Pasteur. And one of the things that I'm really excited about is to really be able to show this framework for large-scale manufacturing. So I'm going to skip to tell you a little bit about malaria. There's lots of tools and technologies that I would love to spend more time on, but we'll do that over coffee or tea. Let me just say a word about mosquitoes and talk about malaria. Actually, no, let's just talk about menstrual pads for 10 seconds. During COVID, one of the things that we started thinking about is, could we do distributed manufacturing of an N95 mask? And we invented this quite bizarre method that you could take a cotton candy machine and you can convert it, and we showed in an entire factory pipeline that you can make an N95 grade material out of a modified cotton candy machine. If you think about the physics of cotton candy, you'll soon realize that it's actually the same physics of these electrospinning-like fiber formation. And one of the things we wanted to do is explore where are the other sets of opportunities in these nonlinear spaces. And something that hit us at that time is actually the lack of menstrual pads. One of the challenges that we face is cotton is not going to be sustainable at the scale, thinking about the sets of temperatures that are changing. It's the most water-hungry plant. Most sets of menstrual pads and the absorptive materials that are made in them are actually made using polymeric materials. It's very difficult to dispose. And we stumbled upon this beautiful, literally, weed. It's called sisal. This is a photograph from Kenya. And we had this aha moment of what if, and by the way, if any of you have had tequila, you've had an agave. It's the same family of plants. It grows all across the world. And we were able to show, by inventing a very simple process, to go from there to a material that now beats cotton at its game. It's the highest absorptive material, literally, that grows anywhere in as kind of a climate that you care about. So we're very excited about manufacturing of this as a material. And again, going back and forth between, right now we're setting up a plant in Kisumu to actually be able to scale this at scale. Okay, let's say a few words about mosquitoes and malaria. So I have a friend who did this calculation for me. It's, you guys can guess. There's a lot of mosquitoes on this planet. I know some of us study them, some of us hate them, but we have no idea what they actually do out there. Like, our surveillance systems for mosquitoes are just absolutely abysmal. If I was to ask you if there was a mosquito flying around in this room and what species it was, it would take you a while. You catch it, you take it to the lab. If you have the right taxonomist, they might be able to figure it out. We started thinking about this problem, primarily trying to help this poor postdoc from Australia with a little tweezer in his hand and trying to detect the species of a mosquito one at a time. By the way, Ronald Ross did exactly this when he discovered kind of the context of malaria. And I'm going to play a sound, and I want you to tell me, what are you hearing? First of all, can you hear something? Can anybody shout out, what are you hearing? So I'll pass this. This is the deadliest orchestra on the planet. It's the 40 most deadliest mosquitoes. We went to the CDC. We recorded this, all recorded through cell phones. It turns out mosquitoes mate in flight, and they have very specific frequencies that they talk to each other with, and we were able to train machine learning algorithms to actually detect them. So just with a $5 flip phone that you can flip around, this app is out there. You can test and play with it. We've been building a database of acoustic detection. And there's a parallel version of this where we built a more molecular mechanism for essentially detecting the pathogens that are in single bites for these sets of mosquitoes. This is now a program that we run in several countries. This is Madagascar. These kids are the ones that they are mapping the mosquitoes in their entire village. There is no government official coming. There is no other sets of services. It's just literally built on communities trying to collect the kind of data that's necessary for their health. Also, they're buzzing with excitement. which I really like. Okay, I'm going to switch and talk briefly about microscopes a little more. I think one of the things that I deeply love about microscopy is how versatile it is. And you don't have to believe my word, you can believe ChatGPT's word. A friend of mine did this. It's like, what are the 5 most important tools of science? And it decides to put microscope on number 2. I think that's absolutely wrong. Mathematics should have been number 1. And you know, one of the things that ends up happening is how many people really have access to research-grade microscopes. So we started thinking about this question because the field was moving too slowly, and we ended up kind of in this framework of developing an entire open source ecosystem of hardware and software for any kind of bioinstrumentation more generally. We started with what we call as a platform called Squid. That's one of the instruments. I'll turn this on. This object has a capacity to do real-time ML-based diagnostics of malaria slides. We'll kind of play with it. I'll show you some of the data associated with it. But one of the beautiful things that has happened, although we started with a seed of an idea, it grew to a point where now there are 30 or 40 different types of instruments, all open source. Anybody can build them, the entire software stack. And then we've been starting to grow to build a lot more biomedical capacity in communities to be able to do. So I'm just going to give you a few sets of examples. This is in the context of malaria. So this is Kalahandi. This is one of our field sites in India. And the current assay that they are using are actually the GIMSA stain. But one GIMSA described that in 1904. And we haven't actually changed that, and this is exactly what we're doing. That's Durga right there. He's going to spend 9 to 10 hours on a microscope. I know we all, once a while, spend 9 to 10 hours on a microscope. Doing this day in and day out is extremely difficult. One thing we asked is, I live in San Francisco. We have, and again, I don't even have to tell you all this. There's a ton of problems coming. The diagnostics has a resistance. The drugs are starting to get resistance. And I walk out when I take my kids to school every day. I see a Waymo pass by, completely autonomous. There is no human being, and I kind of shake. It's like, what the hell is going on in my world where why can't we solve these sets of problems? And actually, this is kind of what led with a fantastic graduate student, Hong Chuan, who also happens to be here. We started tackling malaria microscopy in an autonomous manner. This was the original octopi. It kind of grew over the years. It really does take 10 years of your life to really build certain sets of tools, but we're very excited about what we've been able to build. And I will do a live demo for this, so we're going to save it for that. But needless to say, we can at this point in an autonomous manner do a diagnostic test where the system can read between 10 to 20 million cells in a period of 5 minutes. This is data collected in the field. We have many of these instruments in the field now where we're starting to collect the largest digital malaria database. And one of the power of this really is real-time ML. So let's see if I actually have a video of it. If I don't have a video of it, we will just play it. We will do a real demo. But one thing that I'm really excited about right here is if you watch this, the microscope is making decisions about which ones of these things are parasites at the same time it's taking the pictures. So this video that you're watching is completely real time. There's no telemedicine. There's just NVIDIA sets of processors. So thank you, gamers, that have actually made things like this possible, if some of you keep doing that for a little longer. And I think, much of this I would not have thought about that something like this in a real-time context would actually be possible. We've been learning a lot by deploying these sets of tools. So it's kind of become quite a large consortium, for example, of partners. Some of the partners are actually in the room. I don't know if Patrick and Ravi I know is here. Patrick might be around. We're starting to scale this program. So if any of you care about engaging in malaria, one interesting challenge that we came across is some of the smears look like this. How many of you have made a blood smear in your life? So a few of you, it takes time and effort. And in the field, this is what smears look like. No good. We've been making smears for all this time. Nobody asked this question for why they look so bad. I asked some of my friends, could you send me a picture of when you do these smears? And that's the picture I got. This is northeast in India. And literally the smear was made on a dirt road. And we asked ourselves a question, what can we do about this? That led, you've all seen what a smear looks like. You just got to get that right velocity to push through, and that's when you get a perfect smear. And then you try and try and try again, and you don't get it. And, there's a simple solution to it. You can actually just solve the equation, and you can figure out what the right smear is. Anybody could have done this. This is literally the same equation that teaches us how paint dries. So if you've ever watched paint dry, we imaged this kind of a context, figured out experimentally and theoretically what is the right accurate velocity and angle, and that then led to this very simple tool, a 17-cent syringe with an air valve, which is from a bike. We can now build a constant velocity actuator. You put all of these pieces together, that leads to Inkwell, which is a completely 3D printed, a couple of sets of parts. You can build and make as many smears as you want. I'll let this run in the context of, and every single time, for tens of thousands of cycles, we have tested these things. It makes the same sphere. And again, I think it's often, it's kind of ironic. Sometimes I show all the demo of the live malaria diagnostics happening, and then I get, oh, that's interesting, from the doctors that are in the field. And I show this video, and there's like, can I just get that right now? And it's like, you have a 3D printer. The files are online. Go build it. And I think, one of the important things that's been happening is distribution and engagement of sets of peoples to build on top of it has been a very important part of how we think about it. So much of this work we do in an open kind of a context. And what's been valuable has been to think about how do we expand on this. So that was malaria, and I want to end on a little bit on the research side. Something that happened in a really interesting manner. People started coming to us that, could you solve for this? Could you solve for that? So gametocytes came about. We built algorithms to be able to detect gametocytes. And then suddenly there was this student that came by who has a connection in Nepal. He said, no, you need to really build this for sickle cell. It's urgently needed. Our pipelines were packed. We didn't have any expertise in sickle cell. And what we ended up doing is shipping the tool to them, giving them the entire machine learning pipeline, and asked, why don't you do it? These are clinicians. So, and we sent one of the students out there, and within 9 months, I mean, it took us 10 years to get to this point, but now when that has happened, you can build on top of it. Within 9 months, they built these sets of algorithms. This is going to be out on our medRxiv pretty soon. They built an entire pipeline for doing automated sickle cell diagnostics. Same thing is now happening in the context of TB, for leishmaniasis, for many STD diseases. But we're starting to realize that if you enable people to actually engage with the technology and build pieces on top of it, you can create literally almost an app store of diagnostics where they have the ownership and they have a piece of the puzzle. It's their data, their training, and they have a piece of the equity itself in the terms of how the technology gets built and deployed. So it's been fascinating to explore these sets of distribution models as well. And I want to end on the research side of the story. One of the things that we started realizing is as we build these tools and we share them, the number of applications only grow. And it's important to make sure that the people that are engaging, we're starting to train them to build on top and invent other things on top of it. It started for us, we got engaged in a project with the NIH Prevail program for Ebola to implement a codex system out in Liberia. This was one of our first trips. We went out there, did sets of trainings, did sets of work. We learned lots of lessons, but this led to essentially a complete open source codex platforms now to do immunophenotyping. Another sets of examples are around proteomics work, live cell imaging, lots of high content screening, microarrays, Raman, around 20 or 30 of these sets of applications that are all built on this single platform. To just give you a context of this, so this, for example, is now our frugal high content screening platform. A couple $1,000s for the microscope that's completely autonomous, a $3,000 open source robot that picks and places, and this is 5 sets of these things, and you kind of see the data on this. This is the high content data from 96-well screen. Now anybody could actually implement a possible small scale screens for all kinds of novel drug targets. And one of the things that's been powerful has been is the types of partnerships enabled to ask, what is the right sets of questions? So that's happened for a lot of different sets of problems, for example, that we've been playing with. And the joy of all of this has really been engagement with people that I would never had the chance to actually engage or have a conversation. No matter how broad my research program is, I'm not going to be the one that often enough would actually experience the set of a pathogen that we should be applying these sets of technologies to. So this kind of a democratizing as a framework is not just a growth of technology in other people, but it's also your own growth. You kind of start asking bigger questions yourself. You start questioning when you put something out there, what does that actually mean? You start questioning your approaches that you take in science. And I think that's been the biggest lesson for me personally, that not only is it important, it's also intellectually a really incredible experience. It's almost rebirthing yourself and trying to ask new questions that you don't otherwise feel that you are either capable of taking or not. So I know I probably run out of time. I am going to end here. There is 100 other slides, but I know I'm not going to tell you anything about them. I am just looking for a picture that I want to show you. It's kind of fun. There used to be times we've also trained presidents to actually learn how to do microscopy, build their own microscopes. But one of the things that's been a joy personally for me has really been that often enough there are individuals that engage with you that you initially cannot understand their true capacity. I think the one almost infatigable resource in this world is human grit and capacity. This is a photograph from Liberia, and I think you can count how many kids. They're all going to middle school. They are my age, and they're going to middle school. And if you think hard about the history of Liberia, you'll essentially understand why they're going to middle school right now. And that grit is really what powers much of innovation in some sense. But often enough, we build sets of systems that even if people have just an incredible grit to do things, they're still kept out of the system. So I think one of the things that, an appeal that I have to many of you is if you have chances to engage with broader sets of communities, please do. There are lots of networks that we have built that if you want to plug into any of those networks, you can create those sets of networks. We can tell you both not our successes but our failures if you're really serious about implementing these sets of programs. So please reach out, and then thank you again, and I'm sorry to run out of time.
Questions, please use the mic. Those who are online, please use the live chat feature to submit questions online.
I do have a question from online. Okay. Okay. Do you take into consideration sensitivity, specificity, and positive predictive value with your frugal diagnostic tests?
Yeah, I think most of those are in the publications. I didn't get a chance to show this. And I think for the malaria data, the number that we have been looking at, I think machine learning has a really interesting thread that the more we train, the better those sets of numbers get. And one of the challenges that's been on our end is to actually increase the data. I would say currently where we are at between 5 to 10 parasite per microliter. But what I do believe is that these sets of systems have the capacity to go all the way down to a single parasite per microliter. The other thing that we're also implementing in that same program is speciation. And so this is kind of expansion of that program. Much of the demo that I'll show you today is primarily for falciparum. And it's quite fun to watch that demo because you'll kind of get a sense for where the machine makes an error. All the data is visually available, and then we can retrain that because you can annotate those same sets of common errors that you essentially find. So I think with machine learning, what's valuable is that once you've built an engine, as you start feeding it more data, those numbers start to change. And I think for the SnapDX test, those sets of things are also in the published literature. It really depends on the pathogen that we're targeting. And it really, for some of those We do them with and without pre-concentration, so that system has the capacity to also do extractions. And so all of those numbers change. The two test targets that we have chosen right now is one is COVID. That was kind of the classical thing that we built first. But the second one was actually schistosomiasis. And so the first one is in saliva as a baseline, and the second one is a urine sample.
Hi. Thank you. Very nice talk. I like it. My name is Yaovi Kildas. I'm from Benin, and I'm working the NIGRI, the Genome Research Institute. I really want to ask this as a personal question, but I'm not sure I'll be able to meet you because I need to run back to the lab. So I'll just ask it.
If you have to run back to the lab, that's important. Yes.
Yeah. So it has to do with the way of thinking. I really like your mindset, which is, I don't see myself exactly as you, but I wish to go there. But the thing is, what happens is that when you then come into the actual scientific world, you meet the structured way of thinking. There is a way of doing things, and there's a way of writing to be able to secure a grant. And slowly you start moving away from your original desire into fitting into the system. What would you advise for this kind of thing?
Yeah, I mean, I think everybody has their own path. We had at lunch, we literally had an hour-and-a-half long discussion just on this. My very simple kind of an intuition around often is, everybody needs to be able to work in the system that's around you, but we built this system. It's not somebody else that built the system. So we also all take the ownership on our shoulders to change it in the sets of ways that we care about. I mean, going back to kind of just creative expression, the simplest advice I have is have a night science project. Kind of the stuff that you do just for yourself. You don't even have to tell it to anybody in the world. It really is something that you're trying to pursue and explore. And very simple tool like a Foldscope. I think Lax here, several of us are Foldscope kind of in the obsessive category. And one thing that I do often to kind of train myself is for 30 days in a row, I'll challenge myself to observe one new biological phenomena every day. And do that for 30 days in a row. It kind of gets you going in terms of how do you observe this world. And that's not for anybody else's service. It's a little bit just like when you learn to bike or you learn to do a skill. So I think ideas are everywhere. And if you ever feel a little bit that, oh, I'm kind of getting away for the original reasons when I started doing science, you can initiate and start these offshoots. And it just kind of reinvigorates you. And so kind of when I'm out in the field, I'm working in some very harsh conditions sometimes, but I'm also bending on my knees and picking up the insects and the ants and the scorpions that are crawling around. And it just, I think you can live in both these worlds simultaneously. And slowly you have the influence of also changing that direction. So I think it's agency is such a critical part of science, and much of that agency, nobody ever will give you permission to do what you want to do. And I don't think you need anybody's permission. You need resources, but that comes with the convincing power, but you have to build that capacity first sometimes, and that happens with training over time. So I think living in these two worlds is a kind of a way to approach this, and at some point of time you'll feel like I'm willing to now jump from one world to another, and then you have made that whatever pill you take in Matrix. Yes.
I had two questions. The
Yes.
first one is a scientific one, which is sort of your pointer to the loss of biodiversity and whether you think that, there are things that we are missing to identify, detect. And so as you explore more and more, is there a way you can sort of think about getting at that problem? Because you may, by the time you may be able to get to a part of the ocean, there's a part of biodiversity that maybe is at a very low level. So what's sort of your thought process to that?
Yeah, I'm glad. This is not a planted question, but I'm so glad that you mentioned it. We have started to build autonomous tools at this point to get to this question. So one of the tools that we built is called Planktoscope. Now, this is an object. It's a flow-through microscope, $400 in parts, all open source. What it does is you just pour water, and it'll automatically image tens of thousands of cells in that water column. And kind of this is the type of data set that will come out. We have classifiers that will start classifying this and then eventually tabulate every possible thing that it saw. And one of the interesting things about, I think, our capacity to observe is so limited. And one thing that we have been starting to do is, in this context, and especially for the ocean, start building the equivalent of what we call these satellites. Imagine an equivalent of a satellite, but from a data collection point of view. We started with not completely autonomous. So these are sailors that we train. These people love being at sea. At this very moment, there are 15,000 boats in the ocean. And these individuals just love and enjoy being at sea. And we retrofitted their boats to actually do both biodiversity surveillance and also send sequencing samples back to us. So this network is growing now. And we showed that the quality of data that was coming from these boats was equivalent to many of the expeditions, where for a normal expedition, I'll spend $60,000 per day. So if you think your mouse facility is expensive, $60,000 a day, and we go from 30 to 40 days every time. So you burn all your money just being at sea. So we have to rethink how we map biodiversity. I mean, the same goes for pathogens. If we can really crash the price of diagnostics, we're not just going to apply it for humans. We're going to truly do ecological measurements for pathogens. So I think this tension between the two is valuable, and that's what I meant to say, that there is not enough scientists, or as the definition we think about on this planet, to truly map the biodiversity of this planet. We really have to make sure that these tools reach a much broader group of people, and that enriches everybody's lives. It's often enough the microbial world is filled with wonder. And so why is it that you talk about bacteria and stuff to an average person and the first reaction is ew? You know, the first reaction should be like, this is mind-blowing, and I'm looking at my own self in this form. So some of that narrative is starting to change. And I think we don't give people enough capacity, but cost is a huge portion of this. And I mean, I'm super excited about sequencing, but I also know the limitations around, it sometimes make me depressed, and some of the computational biologists might not like what I say. It makes me depressed that we have sequences of organisms that we don't even know what they look like. And that's what happened in the ocean. There was this massive sequencing programs, and they are called just unknown units. And they're really interesting from the sequence, but I have no context of physiology. So that's, I think this is something that we are starting to work. And some of these tools, we are pairing them with genomics as well. So do coupled imaging and genomics.
The second question that I have is that, I'm thinking about, as a biochemist, like your FPLC, your democratized version of FPLC. I mean, there are places in the world where if you could get this in a college, or even in the U.S., there are, institutes that serve underrepresented communities. I'm sure there's not an FPLC in every biochemistry department. But then that's a more specialized equipment than, let's say, a microscope. And do you ever see these things being picked up at a level where they actually truly become accessible, which becomes even relevant, for example, with the increasing cost of research, a constant amount of science funding, and, it can actually make a real difference?
Yeah, I mean, I think I have a very concrete example for this, if these slides were to unfreeze. But when Hong Chuan and I built the framework for squid, what started happening is I started getting emails from MIT and my Stanford colleagues that they need these things in their labs. And I was like, I'm not building for you. Go buy whatever you want to buy. But on the other hand, I actually realized an opportunity there. And we felt that, wait a second, we can both actually have them engage and have them build new applications on top of it. And so this is not I don't want you to think frugal science as a hardware that's for other people and I have a Ferrari. No, these instruments actually operate and outperform most of the traditional tools sometimes because you deeply understand them and you can engineer them to be that specific. So a lot of times in these sets of collaborations, we have been able to build, I mean, there is a confocal that we built on this set of a framework. We can build these sets of tools that allow people to do new things. And that's what matters. So it's not about there are two worlds and we're trying to build. We're just building the whole system from scratch. And everybody should be able to engage. And the only request that we have when people who have technical know-how, if they engage and use the tools, contribute back to the community, add something to that tool, write another computational pipeline, put another fluidic chamber on it, and share that along. So I think I don't see any conflict in it. What I clearly see is a manner where each one of them have a capacity to grow. And, often enough, I'm thinking a lot about access in the US alone. So I don't want anybody to think that these sets of puzzles that I told you about are for some faraway place. I can guarantee you 10 miles from now, or even in your labs, you will find that many sets of constraints hinder what we can actually do. But if you engage in that as a process, then essentially you also have a chance to contribute to the larger dissemination of these sets of technologies that, sometimes the adoption is much slower farther out because we have to build distribution pipelines and other pipelines. But these tools are for everyone.
So with that, let's thank Manu once again. There's a coffee reception in the library where Manu is also, I think, going to do some demos. We have to thank the FAES for kindly sponsoring the coffee reception. And thank you all once again for showing up today.
And then I think we have fold scopes for almost everybody. So if you really care about building your microscope today, join in for coffee, spend some time, and you should be able to put one together.